This video on population genetics is a follow-up to the part 1 video in which I introduced the Hardy Weinberg equilibrium. In this video, I will go through 4 types of problems that might arise in population genetics. These 4 types of problems are listed here, and briefly I will mention them and then I'll go through each individually. So the first type of problem, I might ask you to calculate the allele frequencies, P and Q, and to predict the genotype frequencies if the alleles are in Hardy Weinberg equilibrium. I might ask you to determine whether the alleles of a particular gene in a population appear to be in Hardy Weinberg equilibrium. For certain alleles that are in Hardy Weinberg equilibrium, I will go over strategies to determine the allele frequencies. And then finally, I will go over examples where the Hardy Weinberg equilibrium can be used to estimate the allele frequencies of disorders, even if those particular alleles are not in Hardy Weinberg. So for this first problem, I'm going to do a simulation of the Hardy Weinberg distribution. And so the allele frequencies are known to predict the genotype frequencies, assuming certain conditions are met. So this simulation we would normally do in the class period, and what I'm going to do is I'm going to do this simulation and I'm going to guide you through the first part of the worksheet. You have a choice. You can either use my simulation numbers for your own calculations, or you can find something to simulate this on your own. You can change your allele frequency to be different from the one that I use, and you can then do your own simulation. So I'm going to simulate the alleles in a population using these tokens from a game that I had at home here. And I counted out 15 blue and 35 yellow of these markers. And each one of these is going to represent an allele in the population. I'm going to combine these together in a little cup, and I will perform a simulated mating by randomly drawing 2 alleles at a time from this cup. Before we continue on, let's calculate the allele frequencies for the blue and the yellow alleles. And so the first allele would be the blue allele, and we had 15 of those, and the total was 50. And so 15 out of 50 makes an allele frequency of 0.3. And for the yellow allele, we had 35 of those, and out of 50 total would make an allele frequency of 0.7. Next, I'm going to draw out 2 alleles at a time to simulate the mating. And I'm going to draw out 2 alleles, then I'm going to return those alleles to the population so as to simulate a large population that does not change in size. So each time I draw 2 alleles randomly, I'm going to record what that combination was. And so this first combination was 1 blue and 1 yellow, and so I'm going to record that there. Second combination was also a heterozygote. I'm going to record that again. Third combination was 2 yellows, and so I'm going to record 1 there. Okay, so I completed 25 matings and I got these results. So the blue-blue was 3, the blue-yellow was 13, and the yellow-yellow was 9. So this simulation, the actual genotypes that I observed will go in this number 3. And so this is blue-blue, and I found 3 of those, and blue-yellow was 13, and yellow-yellow was 9. For the rest of this assignment, I'm going to have you calculate number 2 and determine what would be the predicted or expected number of individuals in each genotype given the expected Hardy Weinberg distribution. Then after you calculate number 2, you will use the chi-square statistical test to determine whether to accept these numbers as the same. Next, I'm going to go through an example where you may be asked to determine whether a particular population appears to be in Hardy-Weinberg equilibrium. So here's a population in which there are 2 alleles of the A gene, and there actually aren't any homozygous recessive individuals. And so people might wonder whether the little a genotype is somehow deleterious and maybe it's being selected against. Investigating whether those alleles are in Hardy-Weinberg is able to help answer this question. So the steps that we will go through to solve this problem are first to calculate the allele frequencies P and Q, and then to calculate how many individuals would be expected if those particular alleles were in Hardy-Weinberg, and then we will compare the observed and the expected numbers to see how similar they are. One thing I'll just point out, a mistake that I see students doing a lot is to approach this problem first solving for P and Q and then asking whether this formula can add to 1. And I just want to highlight that this is not the correct approach to determining whether the population is in Hardy-Weinberg equilibrium. You need to compare the observed and the expected. So in this problem, first we would calculate the allele frequencies, and so the frequency of big A would be P, we're going to call that P, and that would be 2 times 90 for the homozygous individuals plus 10 individuals who each have 1 big A allele. And we'll divide that by the total number of individuals, which is 100 times 2, since each individual has 2 A alleles. This turns out to 190 over 200, or 0.95. For Q, that would be the frequency of little a, and for that there are no homozygotes, but there are 10 heterozygotes, and so each of those 10 individuals have 1 little a allele. We would divide that by 2 times 100, and that would give us 10 over 200, or 0.05. Next, we would calculate the number of individuals that would be expected in each genotype if that population was in Hardy-Weinberg. And so the frequency of big A would be P squared, and we're going to multiply that times the number of individuals in the population, which is 100. So that would be 0.95 squared times 100, and that equals 90.25. For heterozygotes, that would be 2pq, and that gives an expected value of 9.5. For the frequency of homozygous recessives, that would be q squared times 100, and that would give a total of 0.25. And so those are the expected values based on our allele frequencies, and we're going to compare those to the observed. So the observed was 90 for big A, 10 for heterozygotes, and 0 for homozygous recessive individuals. And what you can see is that these are actually very close to each other. Therefore, the population probably is in Hardy-Weinberg equilibrium. In order to verify that these numbers are close enough to each other statistically, we would go on to do a chi-square statistical test. For the next example, we'll look at a situation where alleles can be assumed to be in Hardy Weinberg equilibrium. So this example involves the ABO blood type. Blood type is determined by two codominant alleles, which are big IA and big IB, and one recessive allele. The recessive allele is little I. So an individual with type O blood must be genotype little I, and in the United States, the percent of individuals with type O blood is 45%. What we'd like to do is figure out the allele frequency of little I based on the genotype frequency of individuals that have type O blood. So to answer this question, I first want to highlight that we are not going to distinguish between the two big I alleles, the big IA and the big IB. In this problem, we're only interested in the little I allele. So the information that we're given is that the frequency of type O blood, which would be the genotype little I, is 45%, and so we'll call that 0.45. Now, I'd like to highlight that this is not a allele frequency. That is talking about 45% of the population has type O blood, and that is a genotype frequency. So really what this is this equals Q squared. This equals the genotype frequency expected in Hardy Weinberg equilibrium. Now that we have this formula, we can actually solve for Q by taking the square root of 0.45, and that equals 0.67. Next, we like to solve for P, and so you need to remember that P plus Q equals 1. And so P equals 1 minus 0.67, or 0.33. So now we've solved for both Q and for P. And now we can go on to answer the question of what percent of the population is a heterozygote that has at least one dominant allele and one recessive allele. And so the frequency of heterozygotes would be 2pq, and 2 times 0.67 times 0.33 equals 0.44, or 44%. The last example I'll go through is looking at situations where we have very rare disease alleles that are present in the population. These disease alleles are actually often being selected against, and so therefore they would not be considered to be in Hardy Weinberg equilibrium. However, when you have pretty rare alleles, most of those alleles are present in heterozygous form. And therefore, we can actually use the Hardy Weinberg equilibrium to estimate allele frequencies in the population. So the example I'll look at here is the incidence of phenylketonuria, which is an autosomal recessive genetic disorder, and the incidence of this, so a baby is born every one individual in every 10,000. Now, what we'd like to know is what percent of the population are carriers based on this 1 in 10,000 incidence of a homozygous recessive individual. So the 1 out of 10,000 is actually the incidence of getting an individual with a homozygous recessive genotype. That, as we went through in the last example, would be equal to Q squared, or we would say roughly equal, since this allele is actually not in Hardy Weinberg equilibrium because it is being selected against. We can then solve for Q, and so we can estimate that Q is roughly equal to the square root of 1 divided by 10,000, and that will give us a Q value of 0.01. We can then calculate P, so P is roughly equal to 0.99, and at that point we have P and Q, and we can estimate the number of individuals in the population that are heterozygous. That would be 2pq, or 0.02 or 2% of the population would be a carrier for the PKU allele given the incidence of homozygotes.